Anybody else not have these yet? Last week we talked about phylogenetic trees as a way to show the evolutionary history of organisms and classify and organize life according to characteristics that things share or don't. One type of phylogenetic tree is something called a cladogram that specifically focuses on common ancestry. Phylogenetic trees, the way we were doing them last week, sometimes are just based on visible characteristics, like when you made your little butterfly trees or whatever organism you were doing. Cladograms specifically look at evolutionary relationships and common ancestry. So we're going to spend a little bit of time looking at this one as our example. When you look at a cladogram, at a tree like this, one of the things that you can do is you can identify a clade. That's a word that describes a group of organisms that basically includes a common ancestor and all of the species that descended from that common ancestor, okay? On these trees, remember, each node, each branching point represents a common ancestor, kind of the last, most recent species that the two above it are descended from. So when you look at this one, how many clades can you identify in this tree? This is kind of like one of those logic puzzles where it's like, how many rectangles do you see in this picture? Take a look at this for a minute, turn and talk to somebody and count. How many clades do you think you can identify here? I'll give you a hint. Here's one. If we take this common ancestor at the bottom and everything descended from it, the entire tree could be one clade. But there are lots of other clades within this one. See how many you can count. Another way to approach this is to look at how many nodes there are, because remember, every node represents a common ancestor. So we could start at the very bottom and say from this common ancestor and all of its descendants, that is the vertebrate clade. They are all organisms that have a backbone. Or we could start with this common ancestor right here. Everything descended from that common ancestor. That's another one, so that's two. We have a third one right here. This common ancestor and everything descended from it, amphibians all the way over to birds. There's number 4. That would be this common ancestor at the bottom of that orange triangle. And 5, 6. Each of those would be its own clade. So if you said 6, you're right. There's 6 clades that you can identify in this cladogram. Each represents a different group. You can think of that kind of like they're nested inside each other, kind of like the whole domain, kingdom, phylum, class, order, family, genus, species get smaller and smaller and more specific. These clades, the way we just identify them, started really broad and got more and more and more specific. Questions about that? Often cladograms will also include information about shared traits or shared characters. Remember, character means like characteristic. It's that same kind of idea. We divide those kind of into 2 groups relative to whatever we're talking about. An ancestral character is something that originated in an ancestor of the clade and is included in other groups besides just the ones that you're studying. And I'll give you some examples of this in a minute. A derived character is the new one that makes that clade different from everything else. It is important to realize these definitions are relative depending on the clade that you're talking about. So here's an example. This right here, the purple triangle right here that includes primates and rodents and rabbits, is the clade Mammalia. Those are the mammals on this tree. So it says, identify an ancestral and a derived character of the clade Mammalia. So let's do the derived one first, because that's usually easiest to identify. A derived one is the new characteristic, the evolutionary novelty that makes mammals unique. What is the trait that evolved just before this clade that makes mammals different from everything else? Yeah. Hair, right? This one is the derived character for the mammalian clade. They have hair, nobody else does. That's the evolutionary novelty, the thing that makes them different. What are other traits that mammals possess that are ancestral, that other groups also have? There are actually several that you could mention for this. Yeah.
Four limbs.
Basically any of these. So yeah, 4 limbs would be one. That's an ancestral character. Mammals do have 4 limbs, but so do amphibians. An amniotic egg is ancestral. Mammals have it, but so do crocodiles and birds. Bony skeletons, vertebrae, anything that evolved further back in the tree that's a characteristic of mammals, but it's ancestral. There are other organisms that are not mammals, but still have that trait. Here's one for you to try just to make sure you've got those words straight. So now if we're looking at the clade Tetrapoda, so from this common ancestor, amphibians, primates, rodents, rabbits, crocodiles, and birds, turn and talk to somebody next to you, identify an ancestral and a derived characteristic of this clade.
It's after the point. Oh.
So for the tetrapods, what is the derived trait? Why is it 4 limbs and not an amniotic egg? Yeah, so remember we're going from this common ancestor on. So the derived trait is the one that's unique to everything in this triangle. So it's basically the one that came most recently right before. Does that make sense? So all of those amphibians all the way over to birds have 4 limbs. What about the ancestral characters? What could you say for that? Yeah, bony skeleton or vertebrae. Either one of those would be considered ancestral. Questions about those words? So it's relative depending on what part of the tree you happen to be studying at that moment, which are derived and which are ancestral. We also use some words to discuss the groups that we are studying. The in-group is the species or the group of species that we are currently focusing on and studying. In a lot of cladograms, though, you will also include one species that's closely related, but it's not the one that you're studying. It's almost, not quite, but almost like a control group. Like you want something there that has most of the same traits as a comparison. So you can say the reason the ones we're studying are different is because they don't have this. So you can kind of see how they're different from their closest relatives. And we call that closely related species or group an outgroup. So an example of this, an outgroup of the mammalian clade, there's not necessarily just one choice for this. In this case, you could probably choose amphibians as an outgroup. They are closely related. They're kind of the next branch down the tree. They have most of the same traits, but not quite all of them. You could also probably pick crocodiles or birds for the same reason. They're kind of the next branch over on the tree. Does that make sense? So the outgroup is kind of the next most closely related group that's similar but not quite the same. What would you identify as an outgroup of the tetrapods? Yeah, the next most closely related would probably be the ray-finned fish. Yeah.
For the last one, you said that like crocodiles were, but those don't have to diverge before.
The problem with that one is you also have to remember that you can rotate this around a node. So like these two diverged at the same time. So you could, amphibians would probably be a little better outgroup for comparison, but it depends a little bit on the data that you have available. Other questions about in-groups and out-groups in cladistics? All right, so the last thing with all of these trees, phylogenetic trees and cladograms are basically a hypothesis of how you think these species are related. And we talked about this a little bit last week when you were trying to construct your own tree with your little organisms. You make the best guess that you can based on the information you have, but you almost never have all the information. You might be able to look at them, but you might not be able to sequence their DNA. You might be able to study fossils, but not see what their external anatomy actually looked like. You might have DNA samples, but nothing else. And so based on what you have, you do the best you can to compare species and put together these trees, but these trees often act as a hypothesis. A couple of basic principles that scientists use. One, with a fancy name, especially because the point of it is simplicity. I think it's funny that it has a really big fancy name. The principle of maximum parsimony basically means the simplest one is more likely to be correct. You always start with the simplest possible explanation for the facts that you have. So out of these two trees, if we have this data and it shows that there was this mutation from G to a T that happened, and we found it in species 3 and 4. There are kind of two possible hypotheses. Maybe that mutation happened once, and then 3 and 4 diverged after that. Or maybe that exact same mutation just by chance happened to happen in two completely separate instances. This one is a simpler explanation because it says that mutation just happened once, and then it got passed on to both species 3 and 4. So that's the one that we would investigate first. We would try and come up with other sources of data to help back that up. It is possible the one on the right happened, but it's more likely that the simpler explanation is true. Another name for this same principle, this is also called Occam's razor. You shave away any unnecessary complications and keep it as simple as possible. That is more likely to be the correct explanation for what actually happened. Questions about this? All right, so we are going to do an activity now where you are going to use a phylogenetic tree as a hypothesis or to make some hypotheses about the evolutionary relationships of this fossil. Those of you who are at home, I posted this, Kylie and Ashley, I just posted this at home as homework. And so you should be able to open this up. I think I put it on a Jamboard because you need to draw on it today. So what I would like you all to do right now, open the Jamboard if you're at home, grab the packet I gave you if you're in class, read the background information, and then step number 1 on the procedure just asks you to look at the fossil and just based on the observations you can make of this fossil, draw a little mark wherever you think this belongs on this phylogenetic tree and label it with the little letter A, based on what you can observe with the fossil. Then we'll talk a little more in a minute once you have that part done, and we are going to add in some more information and you'll have a chance to revise your hypothesis based on some new information in a couple minutes. The next part of this activity utilizes a database called BLAST. It stands for Basic Local Alignment Search Tool. It's a database of genetic sequences, real-life data from many, many, many hundreds of species that are stored in this database and are publicly available for anybody to use. If you have a DNA sequence, you can also use this to compare amino acid sequences of proteins. You can basically throw your sequence into this website, blast it out into the database, and it will run a comparison with all the other sequences that it has and tell you which ones most closely align. As you read in the background information, we were able to recover a few little snippets of DNA from this fossilized creature. And so we are going to use the nucleotide version of BLAST to see if we can get a little more information. So here is our first DNA sample that we were able to recover. It has all been sequenced. You can see it's nice and long. You don't need to know all of that. But I'm just going to copy and paste that into this little search box. And we scroll down to the bottom and we click BLAST. Sometimes it takes a few seconds for this to run because this is actually searching a real life database with lots and lots and lots of information in here. And then it comes up with your results. So if we scroll down here, you can see it gives us a couple numbers. The query cover just shows us how much of our sequence it was using. So for most of these, it'll be pretty close to 100%. This column over here tells us the percent that was identical. So for the next question in your lab, it asks you which organism most closely matched and what the percent alignment was. This one was 99.98% identical. It's called Gallus. And so if we, let me see, if I just type that in here, the easiest way is to ask Google to show us what Gallus is. Here you go. This is Gallus. It's a rooster. So now that you know that one segment of DNA from this fossilized creature matched almost perfectly some DNA from a rooster, you have an opportunity to change your hypothesis if you choose. On your same phylogenetic tree, you can put a letter B. Don't erase the A because I want to see how your thoughts change. You can put a letter B in a new place if you choose to move your hypothesis. If you choose to keep it in the same spot, just write a letter B right next to your letter A. So I know you thought about it and said, nope, I still think this is where it is. While you're doing that, I'm going to pull up the next sequence that we're going to blast. Everybody ready for the next one? You have your letter B on there. And so we're just going to repeat this 3 more times. We have 3 more sequences for each one. You just write down the organism that it's closest to and the percent alignment, and then you're going to add another letter. So this next time you'll add a letter C if you want to. So we'll blast sequence number 2. Give it a minute to run. So sequence number 2 gives us 99.88% identical to a Drosophila melanogaster. Do you know what that is? We've talked about these before, not that long ago. It's a fruit fly. So now you can go back to your tree again, add a letter C where you think this should be placed. Knowing what you now know, here's one important thing to keep in mind for this part. Just because it shares a gene with a fruit fly, does that mean it is a fruit fly?
No.
No. Do you share genes with fruit flies?
Yeah.
Yes. So it is important to remember some of these could be genes that have been there for a long time, but it shows you that this is part of its DNA. You should now have an A, B, and C on your cladogram. I'm going to pull up sequence 3. Are you ready for number 3? All right. Oh, this is the hard one to type. Okay, this is number 3. Taniopegia. Is that what it was? Taniopegia. Yep. This one. So this one is a bird called a zebra finch.
Can you, how much percentage is that?
Oh, sorry, I missed that part. This one was 99.43% identical.
Wait, I can't even see what it's called. I'm sorry.
Taniopegia guttata. You don't need to write that. It's a zebra finch. Just so you know, it gives us several species that at least partially match this. The next one down, Lonchura striata. Let's see what that is. So here's another one. There's another little finch-like bird, apparently called a white-rumped munia, that also matches this sequence. There's one down here. Corvus is the crow genus. So this one is a crow. So there are several different types of birds that all share this gene, or very closely. You ready for the 4th? So here's our 4th DNA sequence. That's quick. And this one you can probably guess what it is already. 100% identical to alligator sinensis. What do you think that is? There can be a lot of places that scientific names come from. That one sounds like it's referring, sinensis sounds like sinuses to me, and this is just me guessing. I don't know. But my guess is if you look at an alligator skull, there are probably particularly large or distinctive sinuses, so they named it after that. Sometimes they're named after the location where they're first discovered. So, to kind of wrap this up, does anyone need any other bits of information for your 4 hypotheses? You should now have 5 letters on your cladogram, A, B, C, D, E, your original, and then after each of the 4 DNA samples. So now there are some wrap-up and conclusion questions for you to think about. Obviously, this still isn't absolutely perfect. This is based on the information you have, but now you need to draw the best conclusion you have about where you think this organism should be placed on this tree. And then answer the questions that follow to kind of wrap up this lab. And you probably will be able to finish this up and turn it in before you leave today. Kylie and Ashley, do you have any questions about this?